Hello, everyone. Can you hear me?
Yes, we can hear you.
Great, great. Okay, so I have a few announcements I'd like to go through first. Um, Let me do this. Okay, so first announcement, we are going to have a very busy week next week. So next week we are going to have one exam, which is going to be exam number 2. And that exam, let me share with you the syllabus here to go through this. And let me share screen with you. Okay, so here is the syllabus. So next week we have exam 2, and that exam, we're supposed to take this exam this week. So I'm going to move it till next week. It will be early next week, and it will be like available Tuesday till Wednesday. Okay, that exam covers 4 chapters, 11, 12, and the new one, 16 and 18. We are not going to cover chapters 15 and 17. So 15 and 17 are not going to be covered. Okay, so we have chapters 11, 12, 16, and 18 for this exam. The other thing is also next week we are going to have our first lab practical. Remember, we have 2 lab practical, 1 and 2. So next week we have lab practical 1, and I'm going to do this like very close to the end of the week. So it will be available for you Thursday and Friday. Okay. So this is just to know. So next week, 2 things: exam 2, 4 chapters, 11, 12, 16, and 18, and lab practical 1, and that is going to be starting Thursday and ending Friday. The lab practical is going to be covering whatever labs I already put or posted eCampus. Okay, Blackboard. Okay, I'd like to do like maybe review session next week for the lab practical if you like. We can arrange this so we can go through the labs, different labs, and see if you have any question for any particular lab or any particular essay. The nature of the test of the lab will be you are not going to be requested, or you are not going to be asked to write down like an experimental essay. You are going to see some data, some results, and you need to interpret it for me. Like say for gram staining, for example, I will show you like bacteria was tell you that was treated with for gram stain, and I will ask you what kind of reaction they have. Is it gram positive, gram negative? And also I will ask for the shape. Okay, same for capsule staining, same for endospore staining, acid fast. Motility test also will show you something, and I will ask if it is motile or non-motile. And name of the stain used. You need to know when you do endospore staining, for example, what kind of stain. You need to remember those names: malachite green versus safranine versus crystal violet. The decolorizing agent, name of the decolorizing agent, like ethanol, all these kind of things. So it will see some pictures, some figures, and it will be very simple questions. Okay, this is for the lab practical. So it will be very nice and very like straightforward. The other thing is I already posted like an extra credit for exam 1, and that extra credit worth 10 points, and the due date was June 24th. and I believe maybe like 60 or 70% of the class done it for those 10 extra points, but maybe 20% or 20 to 30% of the students haven't done this. So I'm just wondering why you are missing this kind of opportunity. 10 extra point that is going to be added to your test. Okay,
So I was sorry.
yeah. So just always look for announcement, always look for assignments to see and to watch for the due dates for this kind of exams, quizzes, and stuff like this. Don't miss these kind of points. Yes, you
I
have a,
have quick.
yeah, go ahead, please.
Okay, so remember like when we had the last Zoom and this extra credit, you said like do not worry about it.
I said what?
Do not worry about this extra credit. So that's why I didn't do it.
No, that was that one. No, that was another one. That was another extra credit that I posted, and that was extra credit 2, and I didn't put any kind of questions, and then I said, no, don't worry about that one. But the
Well,
extra credit 1 was placed before that one. There was
yeah.
one. Yeah, go ahead. Yeah.
Well, I did the first one, but the second one, like the
Yeah.
one that was due
I'm
on
talking
June.
about the first one. I'm talking about the first one. I'm not talking about the second one. Yes.
Okay.
Yes, it is one. You have only, if you look at your grade book, you'll see only one assignment for extra credit. And this is what I'm talking about. Yeah.
But I have a question.
Yes.
I also didn't do the one that I believe she's talking about, but I only had it's the one that was due on June 24th. That's the one I did not do because I thought that was the one that you were saying not to do. The one that I completed was the one the test your knowledge quiz. I did that one, but I didn't do the one that was due on the 24th because it was under my impression that not to do it.
Okay, let me go ahead very quickly here and see the grade book what I have. Okay, let me do this very quickly and see here. Okay, so let me. Assignments.
Yeah.
Okay, so
the one titled extra credit to be added to test 1, that's the one I did not do because I was under the impression that it wasn't valid.
Yes.
yeah, okay. This is what I'm talking about. Let me do this. Okay. This is
So my grade is
basic.
0 of 10, so I'm probably one of the ones that you're talking about, but I was thinking that we weren't supposed to do it.
No, let me show you. Can you see this screen here?
Mm-hmm.
Okay, great. So if you look at this one here, you can see only you have like 1, 2, 3, 4 quizzes. Okay. One of them is I'm working on it. It will be available, starting tomorrow till next week. I'm still working on this one. Okay. You already done with quiz number 1, quiz number 2, quiz number 3. And then you have exam 1 already done. And then there was an extra credit for test your knowledge. Is what I'm talking about.
Okay.
Okay.
I think we're on the same page then because I completed that one.
Yeah, this is what I'm talking about. There was another one, that was not really, I said, no, don't do it. Yes,
Okay,
I did
perfect.
that. Yeah.
Thank you.
Okay.
Sorry.
So this is what we have only one extra credit posted so far. Okay.
Perfect.
And I will follow with the exam number 2 with another extra credit assignment also to be added to that exam. Okay.
Okay.
Just a heads up to have an idea. Okay, so now, do you have any question? Any question for me for the test, the exams? The due dates for you have two things going on. One of them is the identification of Staphylococcus isolate that I gave you, and the due date I believe is very close to. I think it is like after July. After July 24, I believe. Let me see here what I have. Okay. So for Staphylococcus, Yeah, the due date is going to be July 6th, okay? So that is an important one. So July 6th is going to be the due date for the Staphylococcus isolate. You have 25 points. So the assignment include the sending, you know, the ID sheet for your isolate and a 2-page essay. You need to write down on this isolate some, give you more information about it, okay? And then you have a PowerPoint presentation for students, which is, I already sent you also the information about the PowerPoint presentation. That's the major ones, 50 points. Yeah, student PowerPoint presentation, and that is the due date for this one is going to be July 23rd. And some student, I think I have a couple of students send me an email asking, you know, what they're supposed to do. If you look at the announcement, there is an attachment, and that attachment will give you the isolate that you need to work with, sorry, not the isolate, sorry, the organism that you need to work with, and what you need to include in your PowerPoint assignment. Okay, have you seen that file?
Yes.
Okay, so if you look at it, I have it here. Can you see this page here? Can you see that one? I have here the different kind of microorganism and the name. Each student, what you need, what each student is assigned for. Can you see that one here?
Yes.
Okay, so we have two different kind of microorganisms that we're going to cover, bacteria and viruses. So you'll see your name and you will see the organism that you are working with. Okay, so on your PowerPoint presentation, this is what you need to do. You have a maximum number of slides between 15 and 20 PowerPoint slides. You're not going to present it. You are going to send me a PowerPoint presentation that has this kind of information. Okay. And due date again for this one is going to be July 24th. So that is another thing that you need to put in consideration. Okay. Now, any question for me?
I have one additional question just for the staff ID lab. Within our essay, within that 2-page essay, do we need to answer the questions that were within our PowerPoint about how the test was, how each test was conducted, or should the essay just be further information about our isolate?
Yes, only information about your isolate. When you use those kind of questions to help you, you know, when you put your ID.
Identify.
ID sheet, yeah, it's very simple. You need to fill it for me, you know, based on the test results that you have in the PowerPoint presentation. And then you are going to write down a 2 to 3 page for just about, you know, the isolate, you know, the significance of that particular isolate.
Okay.
It's, you know, kind of virulence factors they have. This is what I'm looking for. And if they are pathogenic to animal or to human or whatever, and how it can be treated. This is mainly that you need to focus on.
Okay, thank you.
Yeah. So any other question for me? Anything in the lab to need to to ask me or to go through, you know, a thing that is not clear? I can make it more clear for you. Anything in like chapter 12 or 11 and 12 for now?
I think I'm good.
Yeah, as I mentioned, that will be very, very efficient if you guys email me questions. So I can prepare those for you and I can go through this, you know, with the class. It will be more efficient to do this. Like for example, next week I'm planning for a review session for the lab practical. It will be more efficient if I received like some questions about the subject, you know, the lab, which lab, you know, that you need to have more information. you know, that will make it very efficient in terms of delivering the information for you. So just put this in consideration, you know. And one more thing, if you look at your grade book and you see some of the lab assignment that is not graded yet, that means I haven't received your assignments. So please do check your grade book and see exactly what's going on. And if you already send it, I need from you to forward me that email that you sent, because I need to check the due date. So if for any reason you see that, look at the assignment and it is not graded yet, but you sent the assignment 2 weeks ago, forward that email to me. Maybe I missed it, within all the storm of emails that I'm receiving. So just email me that email, forward it to me. So I will take care of your the grading of your assignments. I don't need to leave everything to the last minute. So I'm trying very much to have all the grades updated as fast as possible, so you know exactly where you're heading and how you're doing. So bottom line is, this week, expect to see a quiz for 2 chapters, 16 and 17. And next week, it will be a kind of a busy week for you. You have a practical at the end of the week, and you have exam 2 at the beginning of the week.
I have a quick question about chapter 18.
Yes, ma'am. Go ahead.
Okay, so I was reading chapter 18, and then I came over like the topic of the Archaea plastida. Can you go over a little bit about the characteristic of the Archaea plastida a little bit?
Let me see what I have here. Any idea? So you said Archaea?
Yeah, Archaea plastida. I can put on the chat.
Archaea, I am not going, you are not responsible for Archaea. I stated at the beginning of this talk, you are only responsible for chapter 16 and 18, which is diversity of bacteria and diversity of eukaryotes.
Okay.
So you don't need to worry about the Archaea now.
Okay, what about the fungal groups and their characteristic?
Yes, what about it?
Can you go
Those
over
are eukaryotic. Okay, so let me see here. I have this chapter 18. Okay. So those are eukaryotic, and let me see here. Most important thing you need to know about fungi or fungus, the way they are classified. Okay. So let me grab like a slide here. Let me do this quickly. Let me share with you this diversity of eukarya, and let's focus on fungi. So basically, when you classify fungi, you are classifying them based on the sexual spore that they are producing. This is the way you classify a fungi, okay? And the classification of fungi, first of all, you need to know, if you are studying fungi, you need to know some important features about fungi first, okay? This is very, very important. You need to know that fungi are group of eukaryotes, okay? They have true nuclei, they have true organelles. And some of them are unicellular, like yeast, some of them are multicellular, like other fungi, majority of them. And when they are like filamentous or making like multicellular structures, we call this a hyphae. And the growth that you see on a plate or on like a piece of bread or fruits, we call this mycelia, okay? So those are heterotrophic, meaning they cannot make their own food. So they are decomposer mainly. So they can get the energy they need by degrading and decomposing organic materials to extract the energy. Same way like we do, like animals and human. Okay. The most important thing is also need to know that they are more closely related to animals than plants. More closely related to animals than plants, and they have a cell wall like plants, but the composition of the cell wall is different. They have their cell wall mainly made of chitin. Chitin is a polysaccharide, which is like N-acetyl glucosamine sugar. Okay, so they have this feature here. Now for the classification, they are, in this chapter, we are going to see how many groups. You are going to see the focus mainly on the zygomycota and glomerulomycota, ascomycota, or basidiomycota. Those are the main group of fungi that you need to focus on. And all this kind of classification is basically based on the sexual spore they are producing and where you find those spores. Okay, I'm just looking for a slide here to make it kind of more easy for you. Let me go to this. Let me go here and I'll try to find, because the lab presentation have it like in a very simple way for you. It's kind of more informative. Yeah, here. So
Yeah.
let me stop this one and go here quickly. Bear with me, please. And I need to go to this. Okay.
Just a little quick question before you go to the lab. What is the difference between hyphae and mycelia?
So again, the thing that you are looking under the microscope is hyphae or a hypha. This is the filament structure that you see under the microscope. So this is microscopic structures, okay? And those group of hyphae, when they are kind of visible, we call this mycelia, okay? We call those mycelia. So you got it? You got that one? Okay. So the as I mentioned, when you talk about fungi, you do the classification. You have this Zygomycota, and from the name Zygomycota, those are reproduce sexually, and they can form like a zygote kind of structure. And like here, if you look at this structure here, this is Zygomycota. They have this sexual zygospore here. Also, they can reproduce asexually by producing this kind of sac structure. And this is, we call this a sporangia, okay? And those inside structure, we call those sporangiospores. So the sac carrying those spores, we call it sporangia, okay? And when it break down, it releases all those sporangiospores outside, so they can, germinate and produce more fungi. They have the ability to reproduce sexually, and when they don't have something called male or female, by the way, they have something called genotype, different genotypes. And in fungi, we call them plus and minus. So plus and minus meaning they are genetically different. And if they have this kind of features, so they can meet sexually and they can produce a zygospore. This is a very characteristic feature for Zygomycota. Ascomycota, on the other hand, they also produce this kind of asexual spore, but it is not included in a sporangia. It is basically attached and making like sacs, sac made of those spores. You need to know the name of the structure where they are carrying this kind of spores. We call it in Ascomycota conidia. We don't call it spores, okay? We call it conidia. And the structure which is holding conidia, we call it conidiophore. And that conidiophore comes from the hyphae, okay? So the hyphae is basically growing, and it can reproduce asexually and making those conidia, okay? Like in chains, like in sacs, this is Ascomycota. And the reason why I said that, because there is a Basidiomycota, which is kind of different a little bit. Basidium mycota, they can reproduce asexually, and they produce the basidiospore, not the conidiospore. So they call it basidiospores, and it's carried on basidium. And the basidium here is not attached to like the hyphae, because in Basidium mycota, the hyphae is kind of highly compacted and making structures, we call it gills. And from that gills, you can see the basidium and carrying the basidiospores. So it is not like in a sac-like structures, as you can see. It is basically like a cup. The basidium like a cup, and this is the name Basidium mycota. It is a cup fungi carrying those kind of spores. So the main criteria for classification of fungi is basically based on their sexual and asexual spores that they are producing and how they are, where they are located, mainly. And the main difference, again, when you talk about the classification, you talk about unicellular and multicellular, first of all. Unicellular, you're going to separate yeast out from those, and then multicellular will include the rest of fungi. Although yeast also, in some cases, they can make filaments, like Candida albicans, for example. So this is mainly for the mycota. As I mentioned, if you just specify like a specific question, so I can maybe create like few more slides to make it more clear.
Thank you so much.
Okay, no problem. Okay, so anything else?
No, that's all. Thank
Okay,
you so much.
thank you very much.
That was
Yeah, again, as I mentioned, if you email me like some of those questions, and I can make a presentation and I can go through those in more details for you.
okay. Perfect. Thank
Yeah.
you.
Okay, thank you very much. Okay, so again, if you are interested in this kind of Zoom meeting for a review for the labs, please email me. I need to have some feedbacks because I looked at the recording for Zoom meetings and I haven't seen much of students basically watching it. So it will be good if I need some feedback, please. Okay. Okay, anything else?
Yeah, just one more. If we do have the review session for the lab next week, do you know what day it would be on, or would you let us know?
I will email you definitely, but as I mentioned, that I'm going to move the lab practical to be Thursday and Friday. So we are going to have it either Tuesday or Wednesday for the review.
Okay, sounds good. Thank you.
Yeah. Okay. And is it 4 o'clock is good, or you need to shift the timing a little bit?
4 was fine for me.
Okay. I know it's kind of tough for doing this kind of thing online, but
Yeah.
yeah, we're trying to survive. I think it will be okay. So far, everybody's doing a good job, as a matter of fact. Okay. So again, look at your assignments, and if you are missing any of this, like grading, please forward the email that you sent about the due date so I can take care of your gradings.
Definitely. Thank
Okay,
you.
thank you very much, guys. Thank you very much. Enjoy the rest of the day.
Yeah.
Okay, bye-bye.